Good day to everyone joining us, and welcome to today's X Talks webinar. Today's talk is entitled Drug Labeling Studies in Clinical Pharmacology, Recent FDA and EMA Updates. My name is Sydney Perlmutter, and I'll be your X Talks host for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank Celerion, who developed the content for this presentation. Celerion, a global leader in early clinical research services, offers a unique combination of medical expertise, clinical operations experience, and scientific excellence that gives its clients the confidence to make fast, accurate decisions about their drug development path. For 50 years, Celerion has leveraged the latest operational concepts and technologies to execute safety and tolerability, pharmacokinetics and pharmacodynamic studies in highly controlled clinical environments. These include first-in-human dose escalation, drug-drug interaction, cardiac safety, bioequivalence and bioavailability, metabolism and excretion studies, as well as pharmacokinetic evaluations in patients with impaired renal or hepatic function. Celerion offers feasibility, data management, biostatistics, clinical monitoring, and bioanalytical services. Its founding mission is to help its clients get their drugs to market quickly so that they touch the lives of our family, friends, and people in need around the world. And now I'd like to introduce our speakers for today's event. Sabina Payalunga serves as a subject matter expert in metabolic diseases such as NASH and type 2 diabetes, and she has published extensively in these areas. She joined Celerion in 2015 and has held several roles with increasing responsibility. Sabina received her PhD in biochemistry from McGill University in Canada, and her training also includes 3 postdoctoral fellowships. Arnout van Harst is a subject matter expert in Celerion's respiratory research team and works closely with external experts in the field of pulmonology to support the drug development programs of Celerion's clients. Arnout joined Celerion in 2015 and has over 25 years of drug development experience at biotechnology companies and CROs. He received his PhD from the School of Medicine at Leiden University and is qualified as a clinical pharmacologist in the Netherlands. And now, without further ado, I'd like to hand the presentation over to our first speaker. So, Sabina, take it away.
Thank you, Sydney, for that introduction. Good morning or good afternoon to the folks online today. Thank you for joining Arnott and I as we review updates to product labeling guidance documents and share some of the best practices for study designs and conduct. For those of you not familiar with Celerion, we are a full-service clinical research organization with a focus on clinical pharmacology studies. So we specialize in early phase healthy volunteer studies as well as product labeling studies. In addition to clinical conduct, we offer regulatory and protocol writing services, data management, and bioanalytical analysis as well. We work with both large pharma and small to mid biotech, small to mid-sized biotech companies, where we've ranked quite highly over the past 8 years through the CRO Leadership Award. Celerion is a global company with clinics located in Lincoln, Nebraska, Phoenix, Arizona, as well as Belfast, Northern Ireland. We have our bioanalytical labs in Lincoln, co-located with the clinic, and in Zurich, Switzerland. Our PK scientists are based out of Montreal, Canada, and then we have offices in Vienna and Seoul that support external site studies through our global clinical development department. Now, just as a quick introduction to drug development, when a new drug enters into the clinical stage of drug development, it must first proceed through a series of exploratory type studies. And these are phase 1 and phase 2 studies in which safety, tolerability, pharmacokinetics, pharmacodynamics, and early signals of efficacy are evaluated, as well as dose optimization and dose regimen can be established. Then later, larger phase 3 confirmatory studies are done. And during this period, there's a number of product labeling studies that support the drug development program, the new drug application, and even the drug label once the drug is approved. These studies provide information about the potential of drug-drug interactions, drug bioavailability, and the effect of a meal. Also, we could look at the influence of special populations and how the drug is metabolized in these special population groups. Other product labeling studies include a mass balance study and a thorough QT study to evaluate pro-arrhythmia risk. Now, as just mentioned, understanding the drug pharmacokinetics is an important step of drug development. And the PK is the movement of a drug through the body system. And this includes the absorption, the ability of the drug to pass through barriers like the intestinal lining and the lungs, the distribution, how the drug is distributed through the body and the propensity of it to accumulate in certain tissues and organs. The metabolism of the drug is how it actually breaks down, and then the excretion is the rate and the process of drug elimination. Now, these pharmacokinetic properties can be altered by extrinsic factors like food or even the co-administration of other drugs. And this is typically evaluated in healthy volunteer type studies, and it's going to be the focus of today's talk. Now, there are also intrinsic factors like age, renal impairment, hepatic impairment, genetic polymorphisms, and SIPs that can also affect drug metabolism and PK. These are often done and evaluated in patient studies and special population groups, or this data could potentially even be modeled. Now, the FDA and EMA have updated or finalized several guidance documents pertaining to product labeling studies over the past 4 years, and you can see a full list here. I do want to note that the ICH guidance covers the regulatory regions covers several regulatory regions, including the US and the EU, where these guidance documents have been adopted by the FDA and EMA respectively. Also, the FDA publishes a list of guidance documents that they're currently drafting or reviewing. So we're anticipating an updated hepatic impairment PK guidance coming out maybe later this year. Also in 2022, the FDA held a workshop on obesity bridging PK studies. So there may be a new guidance coming out on this as well. But today we're going to take a closer look at some of the updates made to the mass balance guidance, the DDI guidance from both the FDA and ICH, as well as food effect and acid reducing agent guidance. And during this presentation, we'll share some tips and best practices for conducting these studies. And this comes from a wealth of experience. Over the last decade or so, we've done nearly 100 ADME studies, over 100 food effect studies, and close to 400 drug-drug interaction PK studies. Now, first, I'd like to begin with the mass balance ADME study. And in the human mass balance ADME study, a radioactive drug is administered to track the absorption of the drug. So we look at the concentration time profiles. We could evaluate the distribution in plasma and whole blood, as well as identify metabolites and evaluate the route of elimination. Liquid scintillation counting is applied for the radioactive doses between 50 to 450 microcuries. But when we do micro tracer type studies with lower radioactive doses, then we would look to using AMS analysis. And the results from this ADME study can inform the next steps in drug development. So really give you an indication whether you're going to need to run a renal hepatic or hepatic impairment study, whether certain drug-drug interaction studies are needed, the bioavailability of the drug, as well as this data could be used in PBPK modeling for simulated trials. So the recent FDA guidance recommends that an ADME study be done for all new drug or all new molecular entities, but there are a few exceptions here, and that would include if if there is available literature sources or already approved drug label, then an ADME study is not needed. Also, if the drug is a biologic, then an ADME study is also not required because antibodies, peptides, hormones, for example, they tend to be eliminated through catabolic processes. An ADME study is also not needed if the drug is mainly excreted unchanged through the kidneys, or if it's a topical drug with no systemic exposure. Now, an ADME study is run in a specialized suite with a radioactive license under the supervision of an authorized nuclear pharmacist. It's typically a single-dose study, and the most common label we use is C14. However, we've worked with tritiated drug labels as well. Overall, these are done in healthy male volunteer participants where whole blood, plasma, urine, and feces are collected. Daily liquid scintillation counting is done to support the release criteria. And per the FDA guidance, the release criteria should be that at least 90% or more of the radioactivity is recovered in the urine feces, with less than 1% being collected in less than 1% of the radioactivity found in 2 consecutive collections. For the metabolites, we can measure that in our Zurich lab. One of the main outcomes of an ADME study is the radioactive PK profile. And in the graph, you'll see the total radioactivity is in pink. And this is the sum of the radioactive drug in blue, as well as all of the metabolites that are formed, which are shown in black. This can be compared to the PK profile of a non-radioactive drug, as well as any metabolites if they're already known and measured at this point. The total radioactivity can be measured in both the plasma and the whole blood and visualized as a ratio as shown here. And this gives an indication of the cellular fractioning, partitioning, as well as potential plasma protein binding. Now for the mass balance analysis, this can reveal the major route of elimination and is reported as the cumulative and percent radioactivity in urine, feces, and the total of both. So in the graph, you see the percent total radioactivity dose recovered in feces, urine, and total over 168 hours. Now, as I mentioned, ideally you should be recovering over 90% of the label, and if it's not all completely captured within urine and feces, we could potentially look at other excreta like expired air. For metabolite profiling, this is where we look at the abundance and structure, and any metabolite over 10% of the total drug exposure should be identified and studied in non-clinical toxicology models. Now I'd like to walk through a small case study example. This was a study done in our Lincoln ADME suite. This is a study with gefapixant. It's a purogenic antagonist being developed for chronic cough. The non-clinical data suggested that the drug was mainly excreted through the kidneys. So we did an ADME study in healthy adults. We looked at blood, urine, and feces collection, and all the participants were discharged within 7 days of post-dose as they met the discharge criteria. Starting with the mass balance summary, you'll see that 99% of the drug was recovered, 76% in urine, and then 23% in the feces. So this really suggests that the main route of elimination is urine. And then the plasma concentration profiles were quite similar for the total radioactivity, shown here with the pink line, and then the parent drug in blue. Overall, there was a slightly longer half-life with the total radioactivity compared to the parent drug, and the AUC ratio between the two was 0.88, close to 1, but not quite, suggesting that there was minor metabolites being formed. So not shown here, but we did do the metabolite profiling and indeed identified 6 metabolites, but no one was more than 10% by abundance, and all metabolites together were accounted for 14%. So the main takeaways from this study is that gestapaxin is eliminated through the kidneys, and considering the intended population, chronic cough, this would also include older adults, so it really supports the need to run a renal impairment PK study. And just to continue with this story, our global clinical development team did run the study with our external site partners, where we looked at and observed higher exposure levels in moderate, severe, and end-stage renal disease patients. So overall, this suggests that dose adjustment may be needed in this specific population. Now, I'd like to pass the presentation to Arnaud, who will address drug-drug interaction studies next. Thank you.
Thank you, Sabina. Now, first let me briefly introduce the topic of the drug-drug interactions. Over 50% of marketed drugs are metabolized by enzymes such as the cytochrome P450 or the CYPs to make them less active and readily excretable. And CYPs are predominantly found in the liver, but also to some extent in the intestinal mucosa. And together, CYP activity contributes strongly to the first pass effect of drug and have a strong influence on the amount of drug that is overall absorbed. In addition, there are transporter molecules throughout the body, such as the organic anion transporting polypeptides, or the OATPs, and P-glycoprotein, or referred to as PGP, that have an integral role in the absorption, the distribution, also excretion of drugs. And because many drugs inhibit or induce CYPs or transporter molecules, there is a clear potential for drug-drug interactions. So particularly bearing in mind that, that drugs are often concomitantly administered in clinical practice. Now, risks of such interactions are that there may be a decreased exposure to a drug which may go with a reduced efficacy, but there may also be an increased exposure which may go at the cost of safety in terms of generating more adverse effects, for instance. Now, if we talk about drug interaction, we often refer to perpetrators and victims, where the perpetrator really is the moiety that can induce or inhibit an enzyme or a transporter, and a victim is the substrate whose exposure is changed due to the inhibition or induction of a enzyme or transporter by a perpetrator. So now, with that said, with that said, Sabine already alluded to that the FDA issued new DDI guidance in 2020, focusing on CYP and transporter-mediated interaction, while a broader but less detailed guideline is also available from the European Medicines Agency, which dates back from 2012. Last year, however, there was new ICH guidance, draft guidance issued, document M12, which has been also adopted by regulatory agencies like FDA and EMA. And some of the key topics listed in those guidances are shown in this slide. But ICH M12 is a slightly more comprehensive guidance document, that also, for instance, covers in vitro drug-drug interaction studies in addition to clinical drug-drug interactions. But here I specifically want to touch upon some of the design aspects for clinical drug-drug interaction studies, the selection of index perpetrators and substrates, and also the use of cocktails of substrates that are also mentioned in both guidance documents. Apart from the guidance document that FDA has, there also is an accessory table on their website that can help select the appropriate clinical index substrate inhibitors and also inducers. So that table is very helpful and interactive, allowing to search for a specific SIP, for instance, and then provides a list of potential candidates for drug-drug interaction studies. Now, in practice, drug-drug interaction studies are designed as crossover studies, either fixed sequence or randomized, and occasionally they can be in a parallel design, for instance, if a drug has a very long half-life. The study typically is not powered and would recruit maybe around 16 healthy normal individuals. And in terms of the perpetrator, that's usually dosed at the or given at the exposure that is obtained with the highest recommended clinical dose and the shortest dosing interval. In terms of inhibitors, they can be dosed as a single dose, but also multiple dosing can be preferred. For instance, if there's time-dependent inhibition, or if the substrate has a very long half-life. For inducers, it's a slightly different story because they usually need at least 2 weeks of daily dosing to achieve the maximum level of induction. The substrates often are dosed as in a single dose fashion, but that may be different if the drug has auto-inhibition or auto-induction properties. And typically, we would include 14 to 20 blood samples for pharmacokinetic assessments, and the watch-out between the periods should be at least 5 to 7 drug half-lives long. Now here I want to show an example of a combined CYP3A inhibition and induction interaction study for a cardiovascular drug where the non-clinical data suggested that it is a CYP3A substrate. So in this case, we used in part A, we used an itraconazole as an inhibitor of CYP3A, and we used phenidibine as a strong inducer of CYP3A. The study was conducted as an open-label, 2-period study with a fixed sequence, and in period 1, the drug was administered alone. Subsequently, we would take a series of robust PK samples, and in period 2, subjects would be pre-treated either with itraconazole for several days, or in part 2 with phenidibine, and there for at least for 14 days before the study drug would be administered. In the case of itraconazole on day 4, and in the phenidibine part, that would be on day 14. Again, followed by intensive PK sampling. Indeed, this drug was found to be a sensitive substrate, as also illustrated in the graph here and by the pharmacokinetics in the table below. As it is very sensitive, or its PK is very much affected by the presence of CYP3A inhibitors and inducers. And you'll note that for itraconazole, shown on the left, there is a similar change in effect when compared to phenidibine on the right, but they have opposing effects. So specifically, itraconazole on the left had an increase in exposure of about 4.5-fold, while phenidibine decreased the exposure to the drug by 5-fold. And therefore, co-administration with CYP3A inhibitors and inducers may need to be avoided. Now, the guidance document also covers options for cocktail drug-drug interaction studies, which have the advantage of only of allowing several enzymes of interest or transporters to be studied in only a single study. Now there are some criteria for such studies to have a study like that be acceptable as a validated or as a definitive study. So for instance, one would need a validated cocktail to make sure that the substrates themselves, the probes, have no interaction amongst each other. An example of a validated cocktail is the Cooperstown 5+1 cocktail, and there are several variations to this theme, but also other cocktails are available. Otherwise, the design of a cocktail study is fairly similar to a regular drug-drug interaction study, with the cocktail of probes being administered in period 1, and in period 2, subjects will be pre-treated with the study drug as a perpetrator before the cocktail of substrate is administered. Now, this is a case study that we conducted for a study that for study drug that was shown, and also its metabolites, to potently inhibit or moderately inhibit quite a series of CYP enzymes. So the objective of this study was to evaluate the effect of the study drug on a selection of 5 probes that reflected the most prominent CYP enzymes. And in this study, 20 healthy subjects were enrolled, and substrates were concomitantly administered as a 5+1 probe, where +1 stands for the vitamin K that was added to ablate the pharmacodynamic effect of warfarin. And on the left in the table, you can see the different probes and the corresponding CYPs that they would be substrate for. Now, the cocktail was dosed on day 1 without study drug, but also on day 11 and 30 minutes after the last dose of the study drug. And here you can see the results from this study, where there was really no effect really on CYP1A2 with the caffeine probe, but a weak inhibition was shown for CYP2C9 when warfarin was used as a probe, but quite some moderate inhibitions were found for CYP2C19, 3D6 and 3A4 when omeprazole, dextromethorphan, and midazolam were used as probes respectively. And even with up to 3-fold increase in exposure of omeprazole shown on the left. Now, other types of labeling studies for which new or final guidance was issued are food effect and acid-reducing agent studies. Intake of food can interfere with the absorption of orally dosed drugs, and similar to drug-drug interactions, a change in exposure or absorption rate can impact the efficacy or safety of a drug. There's a series of mechanisms that underlie such interactions, but the key factors also listed in the guidance are that food may increase the gastric pH and thereby interfere with the solubility of the drug. It may also delay gastric emptying and thereby increasing the residence time of the drug. And it may also lead to an increase in bile salts that may sequester the study drug. So it's important to assess food effect early in clinical drug development, as this will also guide further clinical studies during its development. The EMA guideline that was issued in 2012 does touch upon food effects, but not in great detail. So we'll focus here a little more on the FDA guidance that was issued last year. It's recommended to assess the effect of high-fat meals in the early stage of development, as high-fat meals are most likely to affect the gastrointestinal physiology and therefore also have the largest effect on drug bioavailability. Now, the new FDA guidance also recommends to consider a food effect study with a low-fat meal, in certain cases. So for instance, if a high-fat meal had a great impact on toxicity or on efficacy, and also in the case where patients may not tolerate a high-fat diet. A pivotal food effect study may still be required at a later stage of development. For instance, if the to-be marketed formulation of a drug slightly differs from the formulation tested in the pilot study, or to confirm a clinically relevant finding from an earlier study. And in terms of meals, there will be some examples in one of the cases that I will show shortly hereafter. But how is a food effect study designed? It typically is a randomized single dose and 2 or 3 way, depending on how many formulations are being tested or if different meals are being evaluated, and crossover study. Treatments are typically the fasting arm, which means that intake of the study drug occurs after an overnight fast. And in the fat arm or arms, there will be a meal given 30 minutes prior to the administration of the study drug. Water intake is usually restricted, and apart from the planned meal as per protocol, there should not be any food intake up to 4 hours after the study drug has been administered. Now, food effect can be assessed by means of a pilot study already in the very early stage of drug development during as an additional arm to a single ascending dose study, for instance, evaluating only a high-fat meal. But it can also be, in the case of a pivotal study, be a standalone study, which optionally also includes a part that specifically addresses the effect of an acid-reducing agent. Now, in this case that we conducted was we focused on a new drug for which the target population is restricted to a low-fat diet, and therefore regulatory authorities requested that a low-fat arm be evaluated in this for this drug. So the study that was set up was a pivotal food effect study. So therefore, the sample size of 24 healthy subjects was based on a power calculation. And the study consists of 3 arms, a fasting arm, a low-fat, and a high-fat meal arm. And in the case of the meals, again, the meal was taken 30 minutes before the study drug was administered. And in the table on the bottom, you can see examples of standard meals that can be given on the left for low-fat meals and on the right for high-fat meals. Now, this graph shows the drug concentrations. In red are the concentrations in the fasted arm. In blue, shown the concentrations with the low-fat arm, and in green are shown the concentrations with the high-fat meal treatment. And also some key parameters are listed on the right. But clearly these data show that under fat conditions, both with the low and the high-fat meal, the exposure to the drug is reduced, while the absorption is slightly delayed, as you can tell from the time to reach the maximum concentration, or Tmax. While this difference was statistically significant, the reduced exposure was considered not to be of or not to have any clinical relevance. Now, also with regard to gastric pH, proton pump inhibitors in the gastric mucosa secrete hydrogen ions to maintain a low gastric pH, but their activity is also regulated by mediators like histamine. And moreover, for the treatment of gastric acid secretion, gastric acid disorders, several types of acid-reducing agents, or ARAs, are used that can help increase the gastric pH. So, for instance, antacids which simply neutralize secreted hydrogen ions. There are proton pump inhibitors which directly inhibit the gastric proton pumps. and are also the most potent suppressors of gastric acid secretion. And finally, there are the histamine 2 receptor antagonists, which indirectly antagonize the proton pump activity. Now, changes in gastric pH may again impact the solution of a drug and thereby also its absorption. And in general, for weak-based drugs, there's a potential that the solution is decreased with ARA drugs, and there may be, as a consequence, a loss of efficacy. And the other way, the converse applies for weak acid drugs, where there's a potential increase in the solution that might lead to a high rate of adverse events. Now, unlike EMA, FDA issued specific guidance for ARA interaction studies, where it is recommended also to conduct these studies in early development, in early stage of development, specifically if patients are very likely to take acid-reducing agents, such as in oncology, where about 70% of patients are being prescribed with proton pump inhibitors as acid-reducing agents. For weak-based drugs that are typically at greatest risk of pH-dependent drug-drug interactions, the guidance also provides a framework with questions about drug solubility and the solution profiles that helps assess the risk of a DDI for a new study drug. Now, when designing ARA interaction studies, there are different types of ARAs that can be selected, and that also depends on the exact purpose of the study. In an ARA interaction study, the timing and duration of the actual ARA treatment depends on the type of ARA that is selected. And while histamine 2 blockers are usually administered shortly before the study drug, antacids are dosed concomitantly, but for proton pump inhibitors, a pretreatment of several days is required. Yet proton pump inhibitors, or PPIs, are usually the first choice for these types of studies if one is really looking for the worst case scenario, because PPIs have the strongest and also the longest lasting effect on gastric pH. Now, if a study like this would lead to a clinically relevant interaction with a PPI for a particular study drug, then the label may state that the use of the drug together with PPIs or histamine 2 blockers may be avoided, and the intake of antacids potentially need to be staggered. Alternatively, one can also decide to evaluate other dosing regimens in another study, for instance, to evaluate whether a lower dose of a PPI or staggered dose of histamine 2 blockers would be appropriate. Now, ARA studies are commonly designed as crossover studies, and certainly with a fixed sequence if proton pump inhibitors are used. And typically, but not necessarily, a single dose of study drug and administration under fasting conditions would apply, except for certain situations that I won't detail here. And across the population, the general population, there is quite high prevalence of a bacteria called Helicobacter pylori, which is a bacteria that is also associated with gastric ulcer disease. But because it is known to affect gastric pH, Helicobacter pylori status should also be assessed during screening for any such study. And finally, as for other DDI studies, the study drug and the acid-reducing agents should generally be administered at the maximum recommended therapeutic dose. Now, I want to share two cases in which both food effect and acid-reducing agent interactions were conducted for a new drug. So the first case is involving futibatinib, which is a small molecule and an orally dosed oncolytic drug. And the second case, which I've shown here on the right, is about cabozantinib, which also is a small molecule and also an orally dosed oncolytic. They obviously have a different mechanism of action, but otherwise they share that they are both poorly soluble and also weak base drugs. Because they are orally administered and because their optimal solubility is at a lower pH, an increase in gastric pH may change absorption of these drugs. Now, in both cases, the food effect study was crossed over with a randomized treatment sequence, while a fixed sequence was used for the acid-reducing agent studies. And in each of the acid-reducing agent study, subjects were pre-treated with PPIs for 5 days before the actual study drug was administered. For futibatinib, there was a increase of 11% of the exposure, a decrease of 11% of the exposure found when it was co-administered with food. And also the time to reach the maximum concentration Tmax was delayed. So this is kind of in line with what you maybe would expect for a drug with that is a weak base. Now, interestingly, in the case of cabozantinib, the Cmax and also the AUC were drastically increased, with AUC being increased by 57%. Again, though, the time to reach maximum concentration also was delayed, so it took 2 hours longer to reach the Tmax. So this is surprising and quite different from the case on the left. Now, in either case, the effects that were observed are not expected to have any clinically meaningful impact on efficacy or safety. Now, with regard to the acid-reducing agent studies that were conducted for both new drugs, there were no changes observed in the exposure, in the AUC, or in the time to reach the maximum concentrations. Now, due to time constraints, unfortunately, we can't cover all the recent FDA and EMA guidance updates, but I would certainly like to direct you or your attention to our recent white papers and articles that outline the regulatory updates, as well as some of the best practices for oral contraceptives, therapeutic drug interactions, also for renal impairment studies and for QTc modeling. But in summary, for now, product label enabling studies are to be considered as part of clinical drug development, with ADME studies allowing full appreciation of a drug's pharmacokinetics and its metabolites. Drug-drug interaction studies can address the risk of concomitant medication in general, while the effect of food and the impact of acid-reducing agents on the exposure to a drug, a study drug, may need to be studied in a dedicated interaction study. And with that, I want to thank you for joining us today. And for those interested, we have included a link here to our product labeling resource guide. But otherwise, we're happy to take any questions. So thank you.
Well, thank you both for that insightful presentation. Now I'd like to invite our audience to continue sending in their questions or comments right now using the questions window for the Q&A portion of the webinar. So I've already received a few questions, so I'll start with those. This first question is for Arnout, and that is, for a DDI study with an IMP that is CYP3A4 substrate and also PGP substrate, do you recommend spacing out rifampin induction dose from the IMP administration in the second period?
Thank you, Sydney, for that question. I think this is a case where you would, like in any drug-drug interaction study, you would, in period 1, you would sort of dose the investigational product on its own. And then only in period 2 you would dose rifampin as the inducer. in this case inducing both CYP3A and PGP. So there's no reason to space it out, if I understand the question correctly. So basically, you don't have the induction period as the first period, but as the second. So you will never have a remaining effect of the induction. Maybe one minor detail to add here, talking about rifampin, we presented a webinar earlier this year about rifampin, because currently, even though rifampin used to be the most popular drug among DDI studies, but due to contamination with nitrosamines, rifampin can no longer be used in healthy volunteer studies, so in drug-drug interaction studies. But that's a secondary comment to. But I hope I answered the question.
Thank you, Arnoud. Our second question is for Sabina, and that is, when is the best time to run a ADME study?
Yeah, that's a great question. So an ADME study should ideally be run prior to entering into phase 2 development. This way you can unnecessarily avoid the exclusion of participants and patients with renal function impairment or hepatic function impairment if the mass balance data shows that these are mainly minor routes of elimination. So it would just help increase the inclusion criteria for your study if you have it done before entering into phase 2.
Thank you, Sabina. Arnoud, our next question is for you. Can you use a cocktail if it doesn't contain the right selection of probes?
Yeah, thank you for that question. As I outlined in my presentation, there's a series of validated cocktails that contain a selection of probes or substrates, if you like, that have been established to go well together. So if I understand the question well, you basically want to select several, but maybe not all of the probes from that cocktail, and that's perfectly right, because if you take only 4 out of 5, they still won't interfere with each other's. So that's fine. And actually, if there's another CYP probe that you would want to test as well, but that doesn't go with the cocktail, you can still add that in a second period as a staggered design of the study. So you can all include it in one study, but you need to add the separate one, the additional probe separately. But yes, you can leave out one of the compounds from the cocktail.
All right, thank you. Another question for Sabina. How does a conventional ADME study differ from a microtractor or microdose study? Microtracer, sorry.
Microtracer study, yeah. So the microtracer study, that's when a low radioactive dose with a low radioactive burden, we're talking about less than 500 nanocuries, is administered. And this could be done in absolute bioavailability type study where you would have the cold non-radio labeled drug co-administered with a hot IV microtracer. Or you could administer the microtracers in even a first-in-human and get information about metabolite profiling very early on. The microtracer too can be given with the cold IP at the same time in the same formulation dose for an ADME study, as long as you collect the various excreta to be able to do the mass balance analysis. And then for the analysis, you would need to do the AMS as well as liquid scintillation MS analysis using these various platforms. So that does differ from the traditional ADME study where we would use scintillation counting and have daily access to the radioactive counts through the scintillation counting. With the traditional ADME study, you're using more of a conventional radioactive dose, and then there is that slightly higher burden. Now, for a microdose study, this is when you administer a very small amount of the drug. So we're talking less than 1% of the active IP or less than 100 micrograms of the dose itself. So very, very small amounts just to get a sense of the drug PK This is often referred to as a phase 0 study. It has very minimal safety risks, so it can be done prior to phase 1 type studies, but you don't get any information related really to safety or PD effects because you're dealing with such small doses well below any therapeutic range. So that's how those different types of studies break down.
Thank you, Sabina. An audience member would like to know, for Arnaut, how can you tell whether a statistically different exposure is or is not clinically relevant?
Yeah, no, that's a good question. I think in one of the cases I showed, there was a statistically different exposure after a interaction between drugs. So the statistics tell that the drug isn't bioequivalent in a certain condition with another drug or after intake of food. But whether it's clinically relevant, that's basically determined by the drug profile. So if it has a very narrow therapeutic index, for instance, you would expect that a slight change in the drug's pharmacokinetics would immediately have implications for clinical practice and be clinically relevant. So whether it is clinically relevant really determines on the drug's effect and adverse effect profile, and that cannot be deduced from the pharmacokinetic statistics in a drug-drug interaction study, for instance, as such, but it needs to be considered in perspective of the drug's entire profile.
Thank you. Sabina, another question for you. Do all metabolites need to be measured in DDI studies?
Yes, actually, the answer is no, not all metabolites. You would typically look to see if the metabolite is over 10% of the total exposure, or if it's an active metabolite. Then you definitely need to do the non-clinical analysis in the toxicology studies, and then continue to evaluate it further in drug-drug interaction studies.
Thank you. Arnaut, another question for you. Can you use a clinical index inducer for a particular CYP if it also inhibits another CYP that is relevant for your drug?
Yeah, thank you for that question. It's slightly similar to the previous question we had, but very different as well. So if I understand it, it's really in this case about a drug inhibiting or inducing a particular CYP, but also having other impact on the drugs, on the victim drugs PK. So maybe inhibiting another CYP or PGP, as was the case earlier. In that case, the induction still requires 14 days, but you can, as the guidance also says, you can build in a pause in the dosing of the perpetrator drug. So you will get rid of the perpetrator drug and the inhibitory effect, but the induction effect will remain over the next 24 hours. So in that case, you can really focus on the inductive effect. So if you induce the metabolite, you will get a reduced exposure to the study drug. And if you would assess the effect while there's also still inhibitory effect on the same or another enzyme, you have a risk of underestimating your effect. So it is certainly wise to do that if you anticipate that may be the case. So build in a break of 24 hours after the induction before dosing the study drug.
All right, another question for you, Arnaut. Can clinical DDI studies be replaced by PBPK modeling?
Yes, to some extent you can, as is done in certain cases, you can model certain drug-drug interaction studies. The thing is, to rely on a model, you really need to build a robust PBPK model first for a new study drug, and therefore you need input of clinical PK data, and partly that can be derived from the clinical studies you've conducted so far. But essentially, you will need, or that's mostly the type of data that you will input in systems like that. You will need interaction data from other DDI studies that sort of will help you differentiate between maybe certain metabolic routes and build the model as such. So yes, you can replace some studies, but you definitely need to conduct some, and therefore typically the most prominent CYPs like CYP3A will be studied in clinical studies to collect the most robust data set and make sure that you can take the right decisions later on. Yeah.
All right, we have time for one more question. So for the 5+1 probe cocktail design, is each inhibitor or inducer drug dosed separately, or are all 5 dosed together on days 1 and 11, for example?
Yeah, that's, I think, something I maybe already alluded to in one of the previous questions. But yes, you basically combine the probes literally as a cocktail, as we know as a cocktail when we go out drinking. But this is indeed a cocktail where you ingest or drink all the probes in one go, and indeed you would do that first in period 1 without a perpetrator, and then again in period 2 after you dose also the perpetrator. So all in one go, yes. I think that answers the question. Thank you.
All right, well, thank you both very much for those answers. We've now reached the end of the question and answer portion of the webinar. If we couldn't attend to your questions, the team at Celerion may follow up with you, or if you have any further questions, feel free to direct them to the email addresses on your screen. Thank you everyone for participating in today's webinar. You'll be receiving a follow-up email from XTalks with access to the recorded archive for this event. And a survey window will be popping up on your screen, and your participation is appreciated as it will help us to improve our webinars. Now, please join us in thanking our speakers, Sabine Pajalunya and Arnout van Harst. We hope you found this webinar informative. Have a great day, everyone.